Hello everyone, and welcome to today's topic, bioenergetics. So today we continue our transition into discussing metabolism and metabolic pathways by trying to understand energy. The learning outcomes for today are to understand that high energy compounds release a lot of energy. This release is often conserved in the cell to do stuff with. We want to describe why various compounds are high energy, and so we'll talk about 3 molecules that contain phosphoanhydride bonds, molecules that contain thioester bonds, and redox molecules. Finally, we will predict the flow of a phosphate bond based on free energy or the flow of electrons given reduction potentials. Today's Meet a Scientist moment is Yellapragada Subbarao. He was the first to isolate and discover the function of ATP. And if you kind of fast forward to where we are now, you can understand that this is a pretty major discovery, right? During his PhD. So a pretty cool thesis. He also developed the use of methotrexate as a chemotherapeutic drug. And this is one of the very first anti-cancer drugs used. Let's jump into it, folks. We're here today to talk about energy. And before we can understand energy at a cellular level, we kind of need to understand the molecules that we call high energy molecules. And so we'll talk about 3 today. We'll start with molecules containing phosphoanhydride bonds, like ATP and GTP. We'll also discuss molecules containing thioester bonds, like CoA, coenzyme A. And these first 2 classes of molecules are high energy because they contain high energy bonds. We'll also discuss reduced coenzymes or cofactors, vocabulary that we've already learned, such as NADH, NADPH, and FADH2. Now, we won't really understand why these are high energy until the end of the semester, but for now let's understand that they are considered high energy molecules because the movement of electrons from their redox capability involves energy and creates energy. Let's start at the beginning, folks, with molecules that contain phosphoanhydride bonds, all right? So, up until now, we've kind of talked broadly about how ATP is an energy signal, or it's a high energy molecule. And for example, AMP, if we think back to when we learned about phosphorylase, AMP is considered low energy. It's a low energy signal for cells. Well, full circle, today we should be able to understand why that is. There is a large release of energy when we cleave phosphoanhydride bonds. The molecule ATP is shown to you here on the screen. It contains 1, 2 high energy phosphoanhydride bonds. So if we were to hydrolyze either this first phosphoanhydride bond or the second phosphoanhydride bond, there would be a large free energy release. However, this last bond here is not phosphoanhydride, it is a phosphoester bond. Cleavage of that bond is not considered to be high energy because it does not release this big amount of energy. So here you can see why, if we were left with only 1 phosphate, like there is in AMP is considered a low energy signal because its bond is not high energy, whereas ATP, having these high energy bonds, is considered a high energy signal because it has these higher energy bonds that can be cleaved to release energy. Now let's talk about why these bonds release so much energy. For the 3 reasons listed here on the screen, and on the next slide we'll kind of draw our way through each of them to try to understand what each of these means. First, When we cleave off one of those two phosphates, resonance can increase. And we should understand from organic chemistry that resonance is a stabilizing force. And when we are releasing energy, we're basically getting to a lower energy state that is stable. So, cleave a bond, more resonance, more stable. Second point, the repulsion from adjacent phosphate groups is reduced. Remember, ATP is super negatively charged. Let's look at this molecule, folks. I count 1, 2, 3, 4, a charge of -4. So if we kind of pop one or two of those phosphates off, we're separating that charge. So now some of the charge is here and some of the charge is here. Coulomb's law tells us that if we have less of that charge around, there'll be less repulsion, we'll be more stable. Finally, there's a larger solvation energy for hydrolysis products. That one is easiest explained with a visual aid. But for now, let's keep in mind that if we have more ions, we can have more hydration shells, way back to what we learned on the first day of class. Let's talk our way through each of these three scenarios, folks. Scenario 1, resonance increases. And if you'll forgive me, I'm going to simplify the structure of ATP here. I'm going to draw the base and the sugar kind of simplified as just A, and I'll only draw the 3 phosphates. Okay, let's start. I'm starting by drawing out each of these phosphates. For ATP, and then I'm going to draw what happens when we cleave one of the phosphates off. Okay, so folks, on the left-hand side, I've drawn ATP. On the right-hand side, I've drawn a DP and a phosphate that's been hydrolyzed off. Is there more resonance? Well, let's just draw in some potential resonance arrows, right? We could potentially draw resonance arrows at all of these locations. And let's draw in all of our potential resonance arrows to the structures on the right as well. Okay, now that we're done drawing our resonance arrows, let's just kind of count how many there are on the left and on the right. On the left-hand side, I'm counting 1, 2, 3, 4, 5, 6, 7 resonance arrows that I drew. So I drew 7. What about on the right-hand side? 1, 2, 3, 4, 5, 6, 7, 8. So on the right-hand side, I drew 8 resonance arrows. So you see here we've increased the potential for resonance, the potential to spread that negative charge over a larger area. So on the right-hand side, we should know, since there's more resonance, it's more stable. This is one reason why energy is released when we cleave a high energy bond on ATP. Point number 2, repulsion between adjacent phosphate groups is reduced. So here I'm going to draw ATP even more simplified, with all of these phosphates just kind of drawn as circles here, and then ADP on the right-hand side. So have we reduced repulsion. Well, remember we have negative charge on each of these phosphates. And you can see here on the left-hand side, we have bam, 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 3 negatively charged things right next to each other. Well, on the right-hand side, now we have 2 negatively charged things right next to each other, and the third one has been chopped off and can now float away and not have to deal with this repulsion of 3 things right next to each other. So by kind of chopping off the last phosphate group, we've reduced the number of charged things that are bonded to each other and having some repulsion due to their charge. This is more favorable. It's going to be a lower energy state to have less repulsion, so another reason why energy is released. Finally, folks, there's a larger solvation energy for hydrolysis products. We can again further simplify the drawing of ATP. So I'm just going to draw ATP, and it has this negative charge, right? Then on the right-hand side, I'll draw ADP, and phosphate is often simplified as PI. So here I'm saying I have an increased solvation energy. Why? Because rather than a single ion on the left, I now have 2 ionic species. And so each of these ionic species, let me fix that real quick. Each of these ionic species can perform hydration gels and participate in all of these lovely ion-dipole interactions, right? So the solvation energy is increased when we hydrolyze ATP because we have more ions for water to interact favorably with. And these are the 3 reasons why ATP releases so much energy when we hydrolyze a phosphoanhydride bond. Up next, we're going to think a little bit about the conservation of energy and what happens in cells when we release energy or when we need energy.